All right, hello. So welcome back to another lecture. So in this lecture, we're going to be looking at histology. So this is chapter 3. So last chapter we looked at cells, and so building on cells, whenever we have multiple different types of cells, right? When we have multiple different types of cells that are working together for a common function, and they're working together, we're going to call that structure a tissue, right? So if you remember from our first chapter, we talked about this level or in this hierarchy of organization with atoms and molecules and then cells and then tissues and organs and so on. The tissue level of organization was just beyond, just a level more complex than that of cells. And so this idea of histology is the study of cells, right? So the study of tissues. So we're going to be looking at different classifications of tissues, and these are going to include epithelial tissues, connective tissues, muscle tissue, and then nervous tissue. So we're going to start with epithelial, we're going to move on to connective, and then muscle, and then we'll finish up with nervous. All right. So when we look at epithelial tissues are very common, and when you look down at your hand, or when you look down at your legs, or you look in the mirror, one of the first tissues you see is a type of epithelial tissue, and this is your skin, all right? And so when we do our skin lecture, we'll look at a little bit closer view of the skin. But in general, epithelial tissues are going to be a layer of cells. They can either be one layer or more than one layer. And these are going to be lining some type of opening. They may be in contact with the external environment. So think about the skin on your hand. The skin on your hand is an epithelial tissue. It's in contact with our external environment, right? With that which is around us, okay? Our environment. If you think about the inside lining of your mouth, right? If you run your tongue on the inside of your mouth, on the inside of your cheek, your tongue is in contact with an epithelial lining. When you drink something or you eat something and then you swallow it, that liquid or food that's going down your esophagus is in contact with the epithelial lining of your esophagus, okay? So we have lots of different epithelial tissues, and so what we'll look at how you can classify them based on their shape, based on the number of layers, and then we'll look at where in the body you can find these different types of tissues, all right? So one other important thing to keep in mind is our epithelial tissues have something called a polarity, right? So if you think of the pole, like the poles of the earth, there are two opposite ends, okay? So there's a north pole and there's a south pole. Different poles of a magnet, right? There's a positive and there's a negative. So the poles of our epithelial tissues means that there is a bottom and there is a top. The bottom we're going to call the basal surface or the basolateral surface, and the top part, which is in contact with the external environment, is going to be called the apical surface. All right, and we'll look at different apical specialization. So there are things that you can find on the apical surface that make that specific epithelial tissue unique. All right, epithelial tissues are avascular, meaning they lack blood vessels. So this indicates that they're going to have a difficult time healing, all right? And so we'll see that our epithelial tissues are constantly being replaced by new cells on the underside, and we'll look at this specifically when we do our skin lecture, okay? And then all of our cells are going to be attached to the basement membrane down here, which is sitting on top of underlying connective tissue, okay? So we mentioned that on the apical surface, sometimes you can have what are called specializations, all right? And these are going to be things like microvilli and cilia. Okay, so first, microvilli are going to increase the surface area. So if you look at this cell here, instead of just having a straight, flat apical surface, it has these nice large ridges and these convolutions. These are called microvilli, and they increase the surface area of the cell, and we'll see what increasing the surface area is useful for in just a second. I'll ask you a question about it. The other specialization we have are cilia, all right? So cilia are these little microtubule filaments that extend out from the apical surface of some cells, all right? The cilia is acting to propel things over the surface of the cell. So if you look here, imagine this is the cell here, kind of this tan region, right? These pink, purple kind of hair-like structures, these are the cilia, and then we have some substance, maybe it's mucus or something, over the surface of it. So as the cilia beat and they move, they propel this, in this case, say this is mucus, it beats and propels the mucus along. This is exactly what we see in our trachea with our epithelial lining our trachea that we'll look at later in the lecture. But we're beating and we're propelling the mucus up toward our pharynx, ultimately to our oral cavity, where then you can then swallow it or you can spit it out. In the trachea, this is what's called a mucociliary escalator. So muco meaning mucus, ciliary meaning cilia, and it's an escalator. We're moving it up. So this idea of moving up the mucus by the movement of these cilia. Okay. So when you look at microvilli, why would we want to increase the surface area of a cell? Okay, so think about it, pause the video, try to answer it, and I'm going to move on. So we want to increase the surface area of a cell because we want to increase our ability to absorb or reabsorb something, right? So think about these terms of absorb and reabsorb, okay? They're similar, but they're different, all right? So absorb is when we're doing it for the first time. We're bringing in nutrients, we're bringing in things for the very first time. This is happening in the GI tract, right? So you can absorb some things in the mouth, but you also absorb a lot of things in the small intestine, some in the large intestine, right? So we're bringing in water, we're bringing in glucose, we're bringing in different nutrients, right? Reabsorption is when you do it the second time, okay, or a subsequent time. This is primarily happening in our kidneys, right? So we have things in our GI tract and we absorb it for the first time into the blood. And then when we send our blood to our kidneys, we're filtering our blood, right? So we filter those things out, and then some of those things we reabsorb back. We bring back in to the blood. So this idea of reabsorption is doing it for the second time. So when you have an increased surface area, right? So if we look at these extensions again, when you see these increased surface area on this apical part of the cell, that means we have more room to be able to bring in more things. So whether that's water, whether that's glucose or sodium or amino acids or whatever it may be, we can bring these things in much more than if we did not have them. Okay. All right. So when we start looking at epithelial tissues, we said that you can divide and classify epithelial tissues based on a couple of characteristics. All right. One characteristic is the number of layers. So this is very simple. It either has one layer or it has more than one layer, okay? So if it has one layer of cells, you call it simple, okay? So simple squamous, simple cuboidal. If it has two or more, you call it stratified. So stratified squamous, stratified cuboidal, right? All these different types. Now, there is a unique one, and it is called pseudostratified. So the term pseudo means false, right? Like pseudoscience is fake science or false science. So pseudostratified, the name here implies that it is falsely stratified, or in other words, it looks like it's stratified. It looks like it's more than one layer, but it's really not. Okay, so we'll talk about this in more detail later and how you can differentiate between true stratified and pseudostratified. We'll look at something in particular. Okay, so that's how you can classify epithelial tissues one way, number of layers. There's one layer, it's simple. 2 layers, it's stratified. 2 or more, it's stratified. And then you have that pseudostratified. Okay. All right. You can also classify the tissue based on the shape of the cells. Okay. And this is very straightforward as well. The cells are either flat and thin, so you call them squamous. They're kind of cube-shaped or box-shaped, so you call them cuboidal. Or they look like a rectangle or a column, so you call them columnar. Okay, so when you're looking at these different epithelial tissues, look at a bunch of different cells in the tissue and look at them together and say, okay, what's the kind of average cell shape? Are these mostly flat in appearance? Are they mostly kind of rectangular and columnar? Are they mostly cuboidal? All right. Sometimes cells of a particular shape or particular type of epithelial tissue, they don't all look the same. So if you're looking at a simple squamous here, right? Simple one layer, squamous flat, they don't always all look flat. Sometimes you may have one that looks kind of round, or you may have one that looks kind of cuboidal. But when you look at them as a whole, you'll know that, okay, these, for the most part, these are pretty flat. They look kind of squished, so you're going to call these a squamous cell. Okay, so as we're going along and as you're studying epithelial tissues and you look at different images, and I'll show you some practice images in a little bit, remember to look at all of the cells in a particular tissue. Okay. This also brings up another point of when you're studying tissues and you're studying them particularly to practice identifying a tissue. So look at a picture of it and identify it. The best way to study, or one of the better ways to study, is to look at a bunch of different images of the same tissue type. So look at 4 or 5 different images of simple squamous tissue. Look at 4 or 5 different images of pseudostratified columnar epithelial tissue. Okay, this will help you begin to identify and kind of clue in on specific characteristics that you see across all different pictures of the same type of tissue. Okay, all right, so we'll start off by looking at simple squamous tissue. All right, so simple squamous epithelium, as its name implies, is simple, so we only have one layer, and they are flat, right? They're squamous shaped. Okay, so when you look at them from a side view, they look very flat and squished. All right. So if you think about this relationship of anatomy and physiology that we've alluded to several times in previous lectures, and we'll talk about really for the rest of the lectures we'll look at, the anatomy dictates the physiology, right? So the structure dictates the function. So if you look at the structure of this tissue, it's very flat. It's very thin. There's only one layer, right? So that tells you that, okay, because it's very flat cell, it's very thin, and we only have one layer, because of this thinness of the layer and the cell itself, maybe this is going to allow things to pass through it, okay? And that's exactly what we see, okay? So you find simple squamous epithelial tissues in areas where you expect things to be crossing from one side of the epithelial tissue to the other side of the epithelial tissue, okay? So if you look at the location here, you expect to find them in the alveoli of the lungs, the glomeruli of the kidneys, blood vessels, and different ventral cavities. So if you think of the alveoli in the lungs, right? So the alveoli, this is where we have gas exchange happening, right? So we want to breathe in oxygen, we inhale oxygen, and we want that oxygen to go from the inside of the alveoli into the blood, right? But in order to get into the blood, it's got to cross a barrier, okay? And this is called the respiratory membrane. The respiratory membrane is extremely, extremely thin. It's just one cell thick. It's one cell thick on the alveoli side and then one cell thick on the blood vessel side. So it's really 2 cells. But the alveoli itself, that's just one cell layer thick. And so because it's very thin, the gas is able to, the oxygen and then the carbon dioxide kind of reversely, is able to cross through that cell via diffusion and just pass through it and then go into the blood if you're oxygen, or if you're carbon dioxide, it's going the opposite way, right? So it's going from the blood into the alveoli, and then we exhale that out. Okay, so diffusion of gases, this is a really important one. So you find this in the alveoli. This image here that you're looking at down here in the bottom left, this is an example of an image of an alveolus with a simple squamous epithelium. Okay, you can also find it in the glomeruli. So in the glomeruli, we said earlier about reabsorption. We're filtering things out and then we're reabsorbing it back in. We want those things to be able to come back in easily, and so having a very thin membrane, a very thin wall to the glomeruli, these capillaries, allows for this to happen. Okay, all right. So next one is simple cuboidal, right? So simple cuboidal tells us that this tissue is simple, so we just have one layer, and we have roughly cube-shaped cells, so cuboidal. Okay, so when you look at images like this, you're going to see roughly cube-shaped. Now remember, in schematics like this and in drawings like this, they're all going to look the same. But when you look at them in an actual image, don't expect them to all look perfectly cube-shaped. Okay, so they're kind of box in shape, roughly just as tall as they are wide. Okay, the nucleus is usually right in the middle, and we find these in areas like the you find them in areas like the kidney. You can find them in the thyroid as well. You can find them in the female ovary as well. Okay, so if we look here, kidney tubules, ovary, thyroid gland here. So when you look at this kind of drawing down here, you look at these and you say, okay, most of these are roughly cube-shaped. They're obviously not flat. They're obviously not rectangular and columnar. So you know that this is an example of simple cuboidal. Okay. Most of these simple cuboidal epitheliums are involved in some type of absorption and secretion. Okay, so if you think about these locations here, kidney tubules were responsible here for absorbing and specifically reabsorbing things that we have filtered out, right? Ovary, this is what's going to be surrounding our different ovarian follicles, which then get kind of pushed out into the pelvic cavity. Thyroid, here we are absorbing iodine in order to make our thyroid hormone, and then we're going to be secreting out thyroid hormone. Okay, so think absorption and secretion when you think about simple cuboidal, right? So here you can see absorption and secretion. This is a good example of what we're seeing in the kidney tubule cells, right? So here's a roughly cube-shaped cell, and here we have water coming through, so it's being reabsorbed from here back into the blood. Okay, so this is a really good example of simple cuboidal, right? Okay, we also have simple columnar, right? So this is our last simple. So we did simple squamous, simple cuboidal. Now we have simple columnar. Okay, so simple, we have one layer, and they are roughly rectangular or column-shaped cells. Okay, now when we look at simple columnar, what you'll see, and this will help you distinguish simple columnar from pseudostratified columnar, is the level of the nuclei. Okay, so here the nuclei are elongated, and they're more or less, they're not perfect, but they're more or less in a row. Okay, they're more or less aligned horizontally. Okay, these simple columnar can have apical extension, so they can, or specialization, so they can have microvilli, like we said earlier. They could also have cilia, and they can help with absorption and the movement of things, right? So if we look at their location, if they are ciliated, we can find them in the respiratory tract. This is the mucociliary escalator we were talking about, the cilia beating and propelling that mucus up toward the pharynx and the oral cavity. If they're non-ciliated, they may have microvilli. We find this in the digestive tract, right? So when you think about cilia and microvilli on the surface of cells, on the apical surface of some of these cells, begin to think, okay, where would microvilli come in handy? Where would this be useful to have it? Well, probably useful in the digestive tract, so you can think, okay, probably simple columnar in the digestive tract with microvilli. When you think cilia, think, okay, where would it be useful to have cilia to beat and propel and move things over the surface of the cell? Maybe in the respiratory tract, things like that, okay? Now, you'll also see, here you see some of the structures. You can have goblet cells. Now, goblet cells are not the simple columnar cells. They're a different type, but they're oftentimes associated with them, and they are mucus-secreting cells. So sometimes in the respiratory tract, you'll see kind of a circular, oval-shaped, kind of clear cell embedded within the tissue, and this is an example of a goblet cell. Okay, all right. Now, here's our pseudostratified. Okay, so all the tissues we've looked at thus far, simple squamous, simple cuboidal, simple columnar, are true simple. Okay, they're simple. They're just one layer. Pseudostratified is technically simple. Okay, because it's falsely stratified. It looks like many layers, but in reality, it's only one. So it's really a simple layer, but we're going to call it pseudostratified because it looks like more than one. Okay. Now, the way you tell this apart, so here we did simple columnar, here we're doing pseudostratified columnar. The way you can tell the difference is, there are a couple of ways, but the main way is to look at the level of the nuclei. Okay. So in simple columnar, the nuclei are roughly in a row. They're roughly straight across. Okay. In pseudostratified columnar, the nuclei are all over the place. You have some at the top, you have some at the bottom, you have some in the middle. They're kind of scattered throughout. Okay. This is because in a pseudostratified, which is technically simple, so in a simple tissue, to be simple, all of the cells in that epithelial tissue have to touch the basement membrane. Okay, they all have to be touching the bottom down here. Okay, so all the cells in a pseudostratified are touching the basement membrane. Okay, so if you look at all these, you can follow them down and they all touch the basement membrane. Now, some of them, like this, it may not look like it touches the basement membrane, but it probably goes behind these cells and then touches. Okay, so they're all touching the basement membrane. But what gives it this pseudostratified appearance is not all of the cells reach the surface. Okay, so they're all touching the bottom down here, but they're not all reaching the surface. So that means that some of the nuclei end up higher than some of the other ones. So it gives this appearance of, well, this cell is on top of this cell. So it looks like it's stratified, but it's really not. Okay. And so we find, and if you look at these histological images down here, all these little black purple dots that you see, those are the nuclei. Okay, so you see they're not in a perfect row. They're some are at the bottom, some are at the top, some are in the middle. They're all scattered throughout. Okay, so if you look at this, especially in this image down here to the right, you can see nucleus way down here, nucleus way up here, nucleus in the middle, nucleus way down here. They're all scattered throughout. Okay, now oftentimes the pseudostratified columnar epithelial tissue can also be ciliated. Okay, so meaning it has cilia on the surface. And so if you look at this histological image, you see this kind of border on the top. Okay, these little hair-like structures that you see, these are the cilia. Okay, so you can find these in different glands. You find them in the male sperm tract to help propel and move along sperm, and then you can also find these in the respiratory tract to help um, move up mucus and move up things from the trachea. Okay, so as you get to specific systems later on, when you talk specifically about endocrine system and specifically about digestive and specifically about cardiac and respiratory and all these different ones, you'll revisit these tissues and hopefully you'll recognize them and say, hey, yeah, I remember talking about pseudostratified columnar when we did histology. Now I can relate it to specifically the respiratory system and specifically the trachea and things like that. Okay. All right, so answer this question. How can you tell the difference between simple columnar and pseudostratified columnar epithelial tissues, right? So simple columnar is one layer. Pseudostratified is also one layer, but it looks like more than one layer. So pause the video, try to answer this question, and I'm going to move on. So how do you tell the difference? Well, you can look at the level of the nuclei, right? Usually the nuclei are going to be kind of all over the place in pseudostratified. Some are at the top, some are at the bottom. In a simple columnar, you'll see them more or less all in a straight row. Okay, so here you can see simple columnar here, and then you can see pseudostratified with the cell nuclei kind of scattered all over the place. Okay, all right, we're going to stop here, and we'll pick up in the second part of the video talking about stratified epithelial tissues.